Well, if we still got another minute. Everybody got quiet. Don't want to let you all get started again and then have to shut all you up again. So, all right, so I'm stubbing in for Vicky today. I believe she is in Mountain View, helping John Barros accept his Drake Medal Award. Although I don't think she's doing much helping other than just cheering him on. But that's why she's not here, which I think is a pretty good excuse. So congratulations to John, if you're out there listening. Presumably you're not, since you're getting the award. But today is a great, exciting day because we're going to be hearing a couple graduate students in the program with their research rotation presentations. Kind of at the end of the year, I think most of you are familiar with this whole procedure here, but as a requirement for the dual title degree in astrobiology, graduate students have to go into another lab and learn a whole new set of tools and languages about there's this a different type of scientific problem than what their dissertation is on. And the final step in that research rotation is presenting it to the community. So that's what's happening today. We're going to hear from Sabrina first and then Kim later. So I'm going to step out of the way because it's not about me and turn it over to you, Sabrina. So please go ahead.
All right, sweet. Hello, everyone. Thanks for coming. If you don't know me, I'm Sabrina. I'm a second-year PhD student in the Earth and Space Sciences. And I did my astrobiology rotation in Canberra, Australia, at the Australian National University. Oh no. I knew this would happen. It's because I did the Zoom thing. In Canberra, right down here. And Dr. Jochen Brox's geobiology lab. And so I'm excited to talk about some of that work today, where I got to work with some lipid biomarkers. Okay, so before we go back in time, we're going to start in modern oceans. So our modern oceans are dominated by photosynthesizing phytoplankton, which are the primary producers we have today. They are so widespread in our oceans that you can see them from space, such as in the example here over the Barents Sea in the Arctic, where we have a huge diatom bloom. So these are tiny little microorganisms that have changed throughout time, but the ones we have primarily today are these chlorophyll sea algae, and there were three main groups of them: the coccolithophores, the diatoms, and the dinoflagellates. And these are the single-celled eukaryotic algae. However, our Earth's oceans haven't always looked like this. In fact, they've changed very dramatically over geologic time. So along the x-axis here, along here, we have time starting 4.5 billion years ago on Earth, and here is modern day on the off to the right there. For most of Earth's history, the primary producers have been dominantly bacterial. So these are these tiny little bacteria that we still have today. However, around 800 million years ago, the dominant primary producer shifted into eukaryotic algae cells. And then around 250 million years ago, we see another transition into the chlorophyll sea algae, and these are the kind of algae that we have today. These transitions are very important to understand how the ecosystems have kind of evolved and become more complex over time. There are some hypotheses that believe that because you evolve, you start with such small, tiny bacteria that are only around 1 to 10 microns big. And by the time we get to chlorophyll sea algae, these can be up to 2 millimeters in size, that this kind of growth of our primary producers has meant there is more kind of nutrient dense primary producers, which has allowed the food web to become more complex over time. So if we want to understand these transitions, we have to go to the most recent transition, which happened during this Mesozoic era, this M right here. And so this is where we get our chlorophyll sea algae. And this coincides with a kind of interesting event, the End Permian Mass Extinction, otherwise known as the Great Dying. So this was around 252 million years ago, and it was caused by this runaway greenhouse effect triggered by volcanism up in Russia. So along here, this is a nice graphic that you can look at where we have time at 540 million years ago right here, and we have modern day off to the right again here. And on the y-axis of this plot, we see the number of marine families or marine invertebrate families, which we can kind of use as a proxy for diversity. So these 5 adaptations right here are the 5 mass extinctions that we've had during the Phanerozoic. And you can see right here during the Permian, we have the largest drop in diversity in the marine families, where we have an estimated loss of 81 to 96% of marine species. This was caused by the dying off of whatever was at the top of your food web, which created a trophic cascade, which meant that your primary producers, their abundance fell dramatically, and this kind of left a void open potentially for our chlorophyll sea algae to then come in and take over the oceans as the primary producers. So this lets us ask these questions. What if things like mass extinctions are necessary for, or like how life recovers after a total ecosystem collapse event, such as a mass extinction. So to actually resolve this, we need to look at how, and more importantly, when marine life actually shows up after a mass extinction. And for this, we're focusing on the End Permian, and we turn to dinoflagellates. So we're curious right now about dinoflagellates because they're the first type of chlorophyll sea algae to emerge after the End Permian mass extinction. There are many, many different species of these today in our modern oceans. Some have the ability to biomineralize. This means that they can create skeletal parts of them that are either carbonate or silica, and some can even create their own organic skeleton. While many of them are photosynthesizing plankton, some exhibit mixotrophy, which is the ability to both photosynthesize, but as well consume other organisms for their energy. It's almost like if you had a big T-Rex like that, and after a day of hunting out in the world, it still was a little bit hungry, so it stuck on some solar panels and was able to consume energy that way. On top of that, even if you haven't really thought about dinoflagellates that much, you've likely encountered them in many different contexts. They are the photosynthesizing part of modern coral reefs, and they are the organism responsible for these bioluminescent tides we see off the coast of Seattle. They're also very important for many ecological questions, such as in the recent case with Karenia mikimotoi, which has caught a lot of attention in Australia recently after a toxic algal bloom of these dinoflagellates have killed off over 200 types of fish and shark species off the coast of Australia. Okay, so despite them being kind of this powerhouse of the oceans, they are still microscopic. So how do we actually study something that is that small? And there's this general rule in paleontology that you lose a lot of information over geologic time. So if we look at something like the T-Rex, right now we have, the thing we study most are its bones. And so from its bones, you can learn a lot of information, perhaps like about what it ate from the size and shape of its teeth, or maybe how it walked from what its leg skeleton looked like. However, we lose crucial information such as what did its skin look like, or how did they interact as a community. The same rules apply for the microscopic world. And in the case of dinoflagellates, there are two main lines of evidence. There are something we call dinocysts, and these are kind of these skeletal parts of the dinoflagellate. And so you can see they are not the entire dinoflagellate, but just this biomineralized or organic skeleton part. And again, with these, you lose the information about what the soft tissue of this organism looked like. The other line of evidence we have are fossil molecules. And so this is an example here of a molecule we call dinosterol, and over geologic time, you can see it loses some things like this hydroxyl group, as well as changing, but this is what this converts to over geologic time. And so this is a type of lipid biomarker, and every kind of organism will have these lipids. And so these are a very useful kind of tool that we use to study things throughout geologic time. And you might be asking, like, why don't you use a different macromolecule, such as something like DNA, which contains our entire genetic sequence? And that's because DNA preserves very poorly over geologic time. I think the oldest DNA on record is only around 2 million years old, whereas there have been lipid biomarkers found over 1 billion years old, which is very significant. On top of that, one point. So there are so many different types of lipid biomarkers out there. These are, if you're not used to what lipids are, they're kind of things like your finger fat. So they're found everywhere in our cells. But because there are so many kinds, we kind of have to narrow in the question. We have to kind of narrow in our search and just focus on a few molecules in particular. And for this, we wanted to look at eukaryotic lipid biomarkers. So you and I, we are made out of eukaryotic cells, so our dinoflagellates. And in the cell membranes of our eukaryotic cells, we have these phospholipids that form the actual membrane. But then to stabilize those cell membranes, we have some these other lipid biomarkers, these other lipids. And the one that you're most probably familiar with is cholesterol. So here is what a modern cholesterol looks like. And this isn't the only type of like kind of stabilizing lipid in our membranes. There are many different kinds, but this is the most easy one to kind of understand. This will change over geologic time into two main versions. It will have a saturated kind, which is called cholestane, or you can aromatize them to create this molecule called cholesterol. Yes, and so there are very distinct types of these eukaryotic lipid biomarkers for different organisms that you find all over. And with that, we can see what was around Earth at certain periods of time. Aside from looking at questions revolving like evolution, we can use very old rocks to understand how life has originated and evolved on Earth. And alongside that, this kind of technique that we use to study organic molecules such as lipid biomarkers have been of great interest on things like the Viking landing missions, as well as Curiosity the rover, where the sample analysis on Mars has contained the kind of equipment that we use as well to study these like lipid biomarkers. And on top of that, as we get sample return missions, you might have very, very small amounts of sample sizes. And while you might not see macroscopic fossils or anything, these kind of lipid biomarkers are very widespread and therefore a very useful tool to have. Okay, so now that we kind of know what kind of data we're looking for, what does the current understanding of the fossil record of dinoflagellates look like? Okay, so now here we're zooming into just the Triassic. So this is the 50 million years after this End Permian Mass Extinction. And it is generally thought that by the end of the late Triassic, you have these lipid biomarkers and dinoflagellate cysts pretty widespread throughout Earth. However, the early Triassic record of these is a little less certain. In fact, the oldest dinoflagellate evidence within the Triassic is found from just one drill core in Western Australia called Sahel Shoals. And in the 70s, they analyzed this core and they found this kind of dinocyst, which is called Saheladinium autii, which is the earliest occurrence in the Triassic of these dinocysts. They later reevaluated this same core and they did in fact find dinoflagellate biomarkers. However, these were found in rocks of late Triassic age rather than mid-Triassic age. So these two records don't actually line up within the same core. But yeah, this is all to say that this earlier Triassic record is a little less certain. On top of that, there are hypotheses that dinoflagellates could have evolved and been in high abundance much earlier than the Triassic. And so again, here we're looking at time, and each of these points here are samples in which there have been significant amount of these lipid biomarkers found within them. And they correlate these with something called acritarchs. And so acritarchs, as you can see here, are these tiny little microfossils. However, we don't actually know if these are dinocysts. Yeah, so this is an interesting hypothesis, but it is full of issues. The first one of these are that acritarchs are not dinocysts. These are a kind of microfossil. It's kind of a garbage bin term used in micropaleontology for anything that you can't really assign to any kind of group. And so we just do not have enough information to say what kind of organism this came from. The second point on here is that only around 10 to 15% of modern dinoflagellates actually form cysts. There's kind of theories that when dinoflagellates species feel some kind of environmental stress, they then evolve the ability to actually form a cyst as a kind of protection. But we don't know what this kind of ability looked like in the past. And therefore, we ask the question, are dinocysts really the most effective kind of fossil evidence of these organisms? And then finally, the lipid biomarker record is oftentimes full of contaminations. So many of these samples actually are from drill core samples. And when you're drilling rock, you have to use some kind of drilling fluid to both lubricate and cool your drill bits. However, these kind of drilling fluids are in fact hydrocarbons, which oftentimes will contain the very kind of lipid biomarker you're looking for in your rocks. And it's kind of amazing. These biomarkers can actually kind of percolate into your rocks. And so even if you scratch off the surface of some of these, you can still find these contaminants within them. Yeah. All right, so there are two kind of counter hypotheses with the kind of evidence we have right now. The first is that we take this kind of older record of dinoflagellates at face value and we say, sure, not only do dinoflagellates show up in the early Triassic, but they might even go back further into the Paleozoic. The kind of counter hypothesis to this is that dinoflagellates so far, as we know, emerge in the late Triassic and that any earlier information of that could be a product of contamination. And so to actually answer this question, we must find out when exactly in the Triassic dinoflagellates appeared. And this is finally where my work comes in filling the Triassic lipid biomarker for dinoflagellates. So I was lucky enough to work on 3 sample sets from throughout the Triassic and throughout the world. So these were from Idaho, off the coast of Somerset, and from Timor-Leste. This right here is Pangaea. And so we're just gonna focus today on Idaho, and I'll give you a little sprinkling of some of the work done from the Somerset samples. Yeah, but I'm most excited to talk about Idaho because I actually was able to collect these myself. So like I was saying before, these are from the earliest Triassic, so from this period where we kind of don't know if dinoflagellates are around. And we were kind of interested in them because there's this paper published in the year 2000, which reports abundant triaromatic dinosteroids, which are these dinoflagellate lipid biomarkers. However, these samples collected from Wyoming, from this Dinwiddie Formation, were from these drill cores, and so we wanted to go back and kind of reassess if these were an actual signal of these biomarkers. So in October of 2024, me and my lab mate Hannah, we went out to Idaho and Wyoming to collect some of the rocks from the Dinwiddie Formation. And what we're kind of looking at are collecting these recessive layers of shale, as these shales will often best preserve lipid biomarkers. And the kind of new thing we did is that instead of collecting from a drill core, we actually collected from outcrop. And so then you kind of eliminate this issue, at least, of having drilling fluids contaminating your samples. Okay, so collected a bunch of rocks from Idaho and brought them all the way to Australia in my carry-on bag, where they quickly got crushed up into powders like this. When you have these powders, you can then extract all the extractable organic material and separate them out in these columns. And this is where you separate them out by the saturated fraction and your aromatic fraction. Each of these fractions are then run through a gas chromatography mass spectrometer, where you put your little extract on this column and it will go around a very, very long column. And this will separate your molecules by their mass to charge ratio. And so you can imagine if I'm like a tiny, tiny molecule, I'll go through this column much faster. But if you're kind of this big, sticky, awkward molecule, you'll take a lot longer to get through here. And so that's why we end up with our data in the form of a mass chromatogram, where we have time along the x-axis here. So this is about a 110-minute scan. And every time a group of these molecules hits your detector in your mass spec, you get a peak showing up. And so this is just like a total kind of diagram. Yeah, okay. So now that we know what we're looking at, let's look at what we actually find in the Dinwiddie Formation. So the first thing we see here are, this is looking at the specific mass 217, that's your mass to charge ratio. And what we see around this 61 to 66 mark are peaks. Great. What these peaks correspond to are the molecule colostane. And like I was saying before, colostane are an indicator of eukaryotic cells. And you might think, okay, great, that doesn't tell you much, but this is actually a really cool thing to see because most lipid biomarker studies actually turn out that your lipids have degraded away. Above around 100 degrees, your lipids will break down and you don't actually preserve them in your rocks. So to see these peaks was very, very exciting. When we looked at the aromatic fraction of these, we see again this aromatic cholesteroid. These are these red peaks here, and these other peaks correspond to other type of cholesteroid molecules. We see these peaks appear, and they're pretty nice quality peaks. And so this is a great sign, but this is telling us is that, great, you have eukaryotes, which is not surprising in a Triassic ocean. So to actually learn about dinoflagellates then, we look at a different mass, this 213 mass to charge ratio here. And what we're looking at right now is the mass chromatogram from a reference sample from the Somerset coast, which has historically shown these kind of biomarkers. And from around 69 to 70 minutes in, if you do have dinoflagellates or these lipid biomarkers, you should see these 4 peaks. And we get 4 peaks because there's 4 different like configurations or isomers of dinosterine that you can get. And so that's why there's 4 of them. Okay, so now the Dinwiddie Formation, those peaks are missing. So that is telling us that there's actually no detectable dinosterine within this Dinwiddie Formation, which is not what the literature had previously said. And then looking at the aromatic fraction, again, here's our standard reference material. These green peaks here are the aromatic molecule for dinoflagellates called dinosteroid. And when we look at the Dinwiddie Formation, these peaks are again missing. And so this is really cool because this is telling us that there are no detectable dinosteroids or dinosteroids in this sample. On top of that, our lovely palynologist, Clinton Foster, who's online, ran some of these rocks to look at the microfossils. And what he found were plenty of these little acritarchs. And so these are these kind of spiky microfossils. And the acritarchs in these samples are indicative of a saline environment, which is great. It means we're in the ocean. But Clinton found no dinocysts within this sample, which is telling us that great. Our paleontology or like microfossil record lines up with our lipid biomarker record, and we don't have any evidence of dinoflagellates within these samples, which is pretty cool. Okay, and now I'm going to quickly talk about the Watchet Formation. So this is in a different ocean basin of Pangaea right over here. And Jochen had actually collected these rocks last summer from the Somerset coast. And why he was interested in these and why I analyzed them is because this is one of the areas that first kind of established these dinosteroids and dinosteroids as a kind of viable lipid biomarker for dinoflagellates. However, these these initial organic extracts have kind of been depleted over time. And if you want to use them as a great reference material, you have to have more of that rock. However, the issue with that is that we didn't know where exactly these rocks on the Somerset coast were from. So Jochen went out there, collected a bunch of them. And what we see here is that we were able to find a sample or a similar area that does actually have these reference materials. And so on the x-axis here, these are some of the extracts from this original 1993 paper in which we see these 4 peaks. And this is one of the new samples, 24B418, and you see those 4 peaks. And this is kind of exciting because I was like, oh, I'm going to analyze a bunch of samples and find no dinoflagellate biomarkers, but then I was lucky enough to get to work on some that actually did contain them and see what those look like. And the same story applies for the aromatic fraction, in which those peaks are again present in the sample 24B418. However, one of the other things we found, and this is still kind of preliminary, is that samples from the same area, similar area of a older age did not actually contain these lipid biomarker peaks. This is telling us that within a pretty close area, at some point in the rock record, you see significant amounts of dinoflagellates appearing that you can then record their history in the biomarker record, whereas these older ones do not actually contain that information. Pretty cool. All right, I'll start to wrap up here with some major findings. The first kind of cool thing we did is that we looked at this Dinwiddie Formation that had originally said there were evidence of dinoflagellates. We reassessed these rocks and found out that there is no evidence of them in this ocean basin at this time from these rocks, which is pretty cool. And the second part of this is that we found that the Watchet Coast does in fact contain these biomarkers of interest. And we have more of this kind of reference material to use in the future. And yeah, this is kind of a huge project that's being spearheaded by Jochen and his grad student Salome. And they will continue building this out to figure out exactly how and when the evidence of dinoflagellates appears throughout the rock record. And they'll couple this with some of the microfossil work, which is really cool. Yeah, but I just want to say thank you to everyone at ANU. I had an excellent time over there with Jochen, and there's Janet, their lovely lab manager. Here's Clinton, who's online, the palynologist, and Sal, Jochen's grad student. And then thanks to that AB program for funding this, as well as to my advisor, Marjorie, who helped a lot of this happen. And of course, Hannah, my amazing lab mate who came out with me to Idaho and Wyoming to collect these rocks. Yeah, and I'm happy to take any questions.
Thank you very much, Sabrina. We have time for questions.
Yes.
Okay, this might be silly, but you're collecting samples from an outcropping. How do you know that it's not contaminated from just being like exposed to the world?
Yeah, it totally can be. And so that's where you have to have people who really know how to look at this kind of like the information you're getting out of the mass spec. So one of the things you do is like when you're actually collecting, you don't want to touch them with your hands, otherwise you contaminate them with your fingers. And then you wrap them up in like furnace aluminum foil. And that will mean that you're not kind of contaminating them as you travel with them. But it is a good point that you do see contamination in many samples from the world, from like the rest of the world. And so oftentimes if you see kind of weird looking information, you can do inside outside experiments, meaning that when you have your rock, if you Dremel away the surfaces and analyze that as like an exterior section, if you see these same contaminants that are kind of elevated in this exterior part of the rock, then you can kind of tell that it is a contaminant. Yeah, and it's very true that even like a kind of airplane flying over can spew out a bunch of nasty hydrocarbons that will get into your rocks, but there are ways to avoid that. And on top of that, like there are certain biomarkers that are associated with something such as a flowering plant that only appeared after the Triassic, like into the Cretaceous. And so if you see that kind of biomarker, you must be like, okay, well, this is impossible to have this kind of biomarker at this time. So yeah, there's a bunch of different ways to kind of figure it out. And so there was a lot of background work thinking like, oh, is this an actual signal or are we picking up some kind of contaminant? Yeah. Yes.
So I'm assuming the dinosterol is named after dinoflagellates.
They are.
So I'm assuming this is like a steroid that is specific to dinoflagellates, right?
Yes. Yeah.
Do you know what it does?
I think like what does the
Like, why does it, yeah, do you know?
like
what
why is it different?
Yeah,
Yeah, no, it's a good point. I feel like I don't know specifically, other than the fact that it is one of these like membrane stabilizers that is specifically used for in dinoflagellates. And yeah, I think like all kind of eukaryotes or like modern eukaryotes will have things like cholesterol and like ergosterol and like other kinds of stabilizers, but then different organisms can have different types of these. I don't really know the function of them, and I think that is still kind of a question that's up in the air. But yeah, I think there's a lot of studies going on right now too that are trying to look at modern dinoflagellates to actually take that out. And so how they know it's a dinoflagellate biomarker is because they took a bunch of sad dinoflagellates from the ocean and looked at the living thing. And then you can kind of break them down to simulate what it would be like over geologic time. And that's how you know that these are the right biomarker. But yeah, the function is still kind of this like weird, I don't know. Yeah.
Sabrina, super interesting talk.
Okay.
Presumably at some point, since dinosterol and its associated variants are specific to dinoflagellates, presumably it had to evolve in dinoflagellates at some point over the course of their evolution.
Yeah.
Is it not simply possible that you had dinoflagellates without dinosterol?
That is completely, yeah, that is something we talked about quite a bit, which is really interesting, especially when we're looking at, let me pull them up. These kind of peaks right here. So these are the peaks that correspond with the mass 245. And you have these other like methyl cholesteroids. That's just meaning that you have this cholesteroid molecule with one methyl group over here. But it is this question of like, because dinoflagellates have these peaks as well, these peaks in blue, not just these kinds. And so it is totally possible that they could have evolved this molecule later on in time, but it's just like we don't know because modern dinoflagellates do not have that. So if you see these kind of like 4-methyl cholesteroids and everything, they could very well be, but we just don't know right now with like the information we have. But yeah, it is totally like another possibility. The same thing with the cysts is like, was that only a later evolutionary thing and did they evolve much earlier before then? But yeah, it's tricky when you're working with the fossil record. Yeah, but that's a great question.
All right, well, I think we should probably move on here.
Yeah.
So let's thanks Sabrina again for a great talk.
Yeah.
Came up here.
Here's this. Oh.
I think it looks like you're sit there on Zoom, so I guess when you're ready, just go ahead and start.
Yeah, so you said I just turn off video and audio, but everything else is fine.
Yeah, and then if you plug into the projector, it'll come up automatically and then
Oh,
you
I
can share
see.
your screen.
And I also share screen.
that was excellent. Just very good.
Yeah.
Very clear. Follow it.
It takes a second to do that.
Okay. Well, that's doing that. Does it matter if I just share my little screen?
Oh, that's probably easiest.
Okay. And let me know if there's any.
It's good on Zoom.
Yeah, so.
And then, yeah, and you can do like the hiding everything if you click on the more tab,
On the which tab?
the three dots all the way on the right. So you do hide video panel first and then hide media controls.
Okay,
Yeah.
thank you.
Yeah.
All right, hi guys, so I'm Kimberly Sinclair. If you guys haven't met me before, I'm one of David Catling's students, 5th year PhD candidate. So I had the lovely opportunity to do my rotation in the fall at the Institute of Science Tokyo, or Tokyo Tech. I'll talk about this later, but there's a name change in there. And so I was working with Tatsuya Shinori and Natsumi Noda, and my PI was Tomoaki Matsuda. And we were studying liposomal system encapsulation induced by freeze-thaw and the effect of lipid composition. So I want to take you guys on this journey with me. So I went to Tokyo. In case you guys haven't been to Tokyo, this is a map of Tokyo. There's many different neighborhoods. And Tokyo Tech is located right at the boundary between Odaku and Meguro-ku, and this is the campus map. And this building right here is where the Earth Life Science Institute is located, which I just say this because I think a lot of people know ELSI in the astrobiology community, and it might be of interest to people to know that ELSI exists. It's very cool. I'm going to be talking a lot about it. So Tokyo Institute of Tech, that's what it was called before I went. This is some ground pictures, kind of like laying the landscape of the journey. And it was very cool to experience what a campus was like in Japan. The architecture was stunning. And as I mentioned, it was originally Tokyo Tech when I arrived, but then after a few months, it turned into Tokyo Institute of Science because it merged with the medical campus. And so it's very funny this day, all the students were really excited and we went out to take pictures with the nameplate because they had to redo like all the design, and Japan takes design very seriously. So we all had to change our PowerPoints and everything. So it's very funny. And then this was the Earth Life Science Institute, as I mentioned, in that building. They have like astrobiology graduate conference every year. If you want to attend, I highly recommend. It's an amazing astrobiology institute. And I know Vicky said I can like have some takeaway messages about astrobiology in this talk. So I just was going to say it was an amazing experience being here because they have a different perspective on astrobiology where they actually have all the specialties residing in the same building. So you have your astronomers, biochemists, geologists, all in the same building. And so, for example, just within my office, I had a guy who was studying moon impact modeling, and there was a guy who's doing mathematical modeling of protein folding, and there's somebody else, you know, and I could just turn my chair around and ask anybody one of these questions. And so I think that was really cool. And it made me like wish we had that here, but I still love our program, so I'm not complaining. Okay. And then, as I mentioned, I was in Tomoaki Matsuda's lab. His lab, the sort of pitch is simple to complex. They study cell-free synthetic biology and evolution. And this is a picture of his lab group on the right-hand side. And I just have to thank everybody I worked with so much because they were amazing in helping me learn all this new instrumentation and techniques and made me feel at home in Tokyo. Okay, so we are astrobiologists, and I know this is maybe too simple a way to phrase it, but I'm gonna anyway. This is a big question we ask is like, how did life originate? And this is one of many questions, but in comparison, a synthetic biologist is sort of asking, how can we create life in a laboratory? But the commonality here is, what is life? We both have to define life in order to know what we're looking for. And we have had so many debates and seminars trying to answer this question, and I'm not saying this is the answer. I'm just going to put it up there for quick sake. But let's say life is a self-sustaining chemical system capable of Darwinian evolution, and that some of the key components might be an information system such as RNA or DNA, replication, and primitive compartments are really a fundamental part of that, and that's what I spent my research rotation looking at, were primitive compartments. So when we're talking about compartmentalization, we can go from very simple, such as coacervates, to very complex, such as phospholipid vesicles, and intermediate stages being membrane encapsulated coacervates or fatty acid vesicles. So on the more complex side, you have your fatty acid vesicles, shown here on the left-hand side, which have to encapsulate things like your protein, your RNA, your DNA, etc. And the distinction with phospholipid vesicles is you're having that phosphate head with two fatty acid tails. So it's a more complex version of a liposome, and that's what we focused on, was this sort of complex version of compartmentalization. And the research question we were asking is, how did complex modern cells emerge from simple protocells? So if we got all the right components to form a coacervate or a fatty acid vesicle, how do we get it to go from like a phospholipid vesicle to the amazingly complex systems we see today, which on the left-hand side, we have to incorporate all of these things, but not just incorporate them, but incorporate them functionally. So they actually have to function inside of this compartment. So how did the machinery inside of these compartments become more complex, and how did the compartments interact with prebiotic systems, isolating them and ensuring their functionality? In order to study this, we wanted to see if encapsulation could allow for the stepwise incorporation of components, thus complicating the system. So we were looking at encapsulation as a method to increase the complexity of a compartment. Okay, so there were multiple different like sub-research projects within this research project. The first one being looking into a simple molecule encapsulation such as calcine, and then going more complex. So can we encapsulate calcine to, can we encapsulate a protein? Can we then encapsulate an entire in vitro transcription system? And can we encapsulate an entire in vitro transcription and translation system? I'm going to start out with the simple one, and then I have a little bit of time, if I hope I do, to talk about IVT. Okay, so the calcine encapsulation system, if we're thinking about the lab workflow here, you have your Eppendorf tube, and all that's in here is your calcine, which is your fluorescent dye, and your large unilamellar vesicles, or your LUVs. And so a lot of my research rotation was spent making LUVs, which is a long process, and I can teach you it someday if you want to know, but you end up with this tiny little amount of LUVs in your Eppendorf tube. Just to clarify, large unilamellar vesicles in comparison to giant, large are 100 to 200 nanometers versus giant are like 1,000 nanometers. So we were working in that range. Specifically, we really were focused on 100 nanometer vesicles, and so we would do DLS to confirm their size before working with them. Okay, so we have our calcine and our LUVs in solution. And if you add cobalt chloride, it'll actually quench the fluorescence of calcine. And so this is good to know, because what we wanted to do was expose this system to a freeze-thaw cycle, so a freeze via liquid nitrogen dip, and thawing at room temperature for 20 to 30 minutes. And this should encapsulate some of those calcine molecules into your LUV. And then by adding cobalt after, you are quenching only the fluorescence outside of your LUV, leaving the fluorescence inside of the liposome intact. So you should be able to detect if you've encapsulated any calcine, because you'll see some residual fluorescence. So we wanted to see how these different parameters infect this encapsulation, such as the buffer composition, the freeze-thaw condition, and the lipid concentration. So the lipid type, sorry, there should be a second bullet point, which is lipid type, but this was uniform across all the experiments. We changed lipid type. We were especially interested in the phosphocholine, and we worked with POPC, PLPC, and DOPC the most. For context, POPC is the most abundant phospholipid in humans, as well as egg yolks. Egg yolks are used very commonly in biochemical research. PLPC is a major component of egg yolk as well, and DOPC is found in both human cells and egg yolks, but it's known for its high fluidity. So these are all similar and in that phosphocholine group, but they all differ just slightly, and so we want to study how these different lipid types affect encapsulation as well. Okay, so as I said, one of the things we wanted to check on the effect of encapsulation was buffer. And so we tested two different buffers, your PBS buffer, which is a common buffer used in biological research for stable pH and minimal autofluorescence, and our HKM buffer, which is used in protein synthesis because it provides the necessary magnesium, which is absent in PBS. And so that's sort of the distinction we're making between our two buffers. And as I mentioned, we have our three lipid types in all these experiments. So both in our PBS buffer and our HKM buffer, you expose it to a round of freezing and thawing, and it actually just turns orange from freezing. That's not from the cobalt. So I know that's confusing, but it also turns orange when you freeze it. Okay, and so this was our first result, was looking at the difference between encapsulation and PBS and HKM. So again, the POPC, PLPC, DOPC are the different lipid types. And no freeze-thaw is the sample that did not experience that freeze-thaw cycle. So you should not be seeing encapsulation versus after freeze-thaw was exposed to freeze-thaw cycles. So you're seeing an increase in fluorescence due to encapsulation. And so the takeaway from this plot is that we were seeing a successful encapsulation of calcine via freeze-thaw, that the PLPC and DOPC vesicles encapsulated more calcine than POPC, and that the PLPC and DOPC vesicles encapsulate more calcine in the HKM buffer in comparison to PBS. And so this implies that the magnesium is potentially aiding encapsulation for some reason, but also that PLPC and DOPC could be more viable for encapsulation and the increase of complexity in these systems. We also wanted to test if there could be some side effect from aggregation or fusion of these vesicles. So for context, the image on the left-hand side is if your vesicles are individual and floating in free space, but on the right-hand side, you can get vesicle fusion or aggregation, and this could trap some of the calcine without it being truly encapsulated. And so we wanted to distinguish between these two, and our hypothesis was that if you have individual vesicles, you'll see a linear relationship between fluorescence and concentration, versus if you have vesicle fusion or aggregation, you'll see a polynomial relationship between fluorescence and concentration. So we tested this by doing a range of lipid concentrations from 0.1 to 2.5 milligram per milliliter. And again, on the y-axis, you're seeing calcium fluorescence. And so we saw the linear relationship in both POPC and DOPC, indicating that these lipid types are only experiencing encapsulation, while PLPC exhibits a polynomial relationship, indicating it is experiencing effects from aggregation or vesicle size growth. And so that's good to know, is because we don't want to be measuring just aggregation, we want true encapsulation. And we backed this up with DLS measurements. As I mentioned before, we were working with 100 nanometer vesicles. And so if you measure our vesicles before that freeze-thaw cycle and after, you can see this peak is maintained in POPC and DOPC, but in PLPC, you're seeing some size increases into the 1,000 to 10,000 nanometer range. And so that's, again, we're seeing aggregation with that specific lipid type. Okay, we also wanted to see what happened if you change the freeze-thaw condition. So as I mentioned previously, we were freezing via a dip in liquid nitrogen and thawing at room temperature, but we also wanted to test what happens if you thaw it under a controlled setting at 1 Kelvin per minute. And so doing the same experiment, but with this different freeze-thaw condition, we saw it swap, where now we see a linear relationship in POPC and PLPC, but DOPC in green has this polynomial relationship. And so the effect of aggregation isn't just dependent on lipid type, but it's also dependent on the freeze-thaw condition. So obviously it's a very complex system. We're trying to understand what's happening here. And again, we saw this size increase in DOPC and DLS, but not in the other lipid types. Okay, but now let's try even more freeze-thaw conditions to see what happens. So we have 3 different freezing conditions: your fast liquid nitrogen dip, an intermediate 20 minutes at minus 27.5 C, and a slow 0.5 Kelvin per minute freeze over 60 minutes. And we have 2 thawing conditions, which is fast at 10 minutes in a 15 C bath and 30 minutes at 1 Kelvin per minute. And basically, if we shorthand this, fast is FF versus slow is SS. And so that's what you're looking at here, is the results just with DOPC here in these different freeze-thaw conditions. And the big takeaway is that this fast-slow condition, and this is just a color map showing the same data. The much darker red or the much higher peak is showing you're seeing a lot more encapsulation with fast-slow. Versus your slow, and slow-fast are giving you the lowest encapsulation. And so the conclusion here is that if you have a fast freeze and a slow thaw, you'll get the highest encapsulation. So the conclusions from our calcine encapsulation experiments were that calcine was successfully encapsulated through freeze-thaw. The amount of encapsulated calcine was highest in the order of DOPC to PLPC to POPC. And for DOPC and PLPC, the HKM buffer exhibited higher calcine fluorescence than PBS. Whereas under a fast condition, POPC and DOPC showed linearity, while PLPC potentially had aggregation, and that swapped to DOPC having aggregation under the fast-slow condition. And finally, that the fast-slow condition resulted in the highest calcine encapsulation. So I know that was a lot. And then I'm just going to talk briefly, how much time do I have? Yeah, about the in vitro transcription experiments. I think these are a bit more interesting because we're getting into more complex systems here that are more biologically relevant. And for this, we were testing, again, lipid type, but also the freeze-thaw method, the number of freeze-thaw cycles, the size of the vesicle, and as I said, the lipid type. Okay, so this was fun for me because I'm not a biologist, but I got to learn how to do biology, or not biology, but aspects of it. So in order to set up our IVT system, we had to first get pepper DNA, and I'll talk about why we chose pepper DNA in the next slide. But just for context, we had to go through a PCR chain reaction to get our pepper DNA, and we had to run a gel electrophoresis to check that we got the exact right fragment we wanted at 210 base pairs. And for those who haven't done PCR before, you basically mix a bunch of components in a tube and you expose it to your thermocycler, and that's about it, except for it's actually not that easy because it's kind of sensitive and hard to do. So this took me like 2 weeks, but anyway, did that. Okay, done. Then what is an in vitro transcription system? Just for context, in case anybody is not in biology, we're basically trying to have RNA synthesis via a polymerase in like an artificial environment. And so we use T7 RNA polymerase. You could use another polymerase. And you just give it a DNA template, your polymerase, and then your nucleotides and your buffers and such to produce your RNA strand. But then in order to see how did we successfully transcribe RNA, you need to have a way to detect that in an Eppendorf tube. So we chose pepper DNA to get pepper RNA because pepper RNA binds with this fluorogenic dye HBC 530. So when you successfully create pepper RNA and you expose it to HBC, it'll fluoresce in a very similar wavelength to calcine, which means that we can use the same instrumentation to measure it, which was great for me. Okay, so the experimental setup was we had that T7 transcription kit, which again is your like polymerase, your nucleotides, your buffer. We have our pepper DNA, we have our HBC 530 dye, and we have our vesicles. We then expose it to freeze-thaw, trying to encapsulate all those components into one vesicle. We add DNase to degrade any DNA that's residual and outside of a vesicle. And then we incubate it to allow for RNA synthesis to happen and for the RNA to bind to the HBC ligand. And finally, we add RNase to kill any residual fluorescence outside of vesicle that could have resulted. So this was the first result, was that the calcine, so from previous calcine experiments, as I demonstrated, we showed the highest encapsulations with fast freeze and slow thaw. So that's what I used in this experiment. And we saw that DOPC proceeded slightly, but the result was reproducible. So on the left-hand side was our first experiment, and on the right-hand side was our second experiment. And you're seeing in DOPC here, this is our negative control without freeze-thaw, and with freeze-thaw, you're seeing an increase in fluorescence. And so it was slight, but it was there. So we wanted to dig a bit deeper and see if we could increase this. One thing we thought was that 100 nanometer LUVs are actually pretty small. They have very low volume. So if you just do a back of the envelope calculation, it actually shouldn't really be encapsulating all of these components because it's just not a very large volume. And so we wanted to increase the volume available to us. So we then created 800 nanometer LUVs and wanted to see if we got better encapsulation. So on the left-hand side, you're seeing 100 nanometer LUVs, and the right-hand side was a test with 800 nanometer LUVs, and you are seeing greater encapsulation in the 800 nanometer LUVs. And I'm not going to go over the freeze-thaw stuff because it's not important for this. And then the last test we did was the number of freeze-thaw cycles. So if you go through one liquid nitrogen bath and a thaw, doing that multiple times, and you can see that basically you have a slight increase after one cycle, but then it sort of plateaus or even decreases after that. And so this is showing us that a single freeze-thaw cycle results in the highest encapsulation. So our conclusions from IVT were that the in vitro transcription system proceeded slightly in the DOPC vesicles under that fast-slow condition. Also that the 800 nanometer LUVs exhibited higher IVT system encapsulation compared to the 100 nanometer LUVs, and that repeated cycles of freeze-thaw didn't increase IVT encapsulation. This was sort of fun for me to just write up because Vicky was saying we should talk about skills that we learned. And I was just like thinking, what did I learn? And I realized it's actually kind of a long list. So I learned a lot about biochemistry, which I'm not a biochemist. I learned how to do DLS or dynamic light scattering. I learned how to run a PCR, use a nanodrop, do gel electrophoresis, do RNA synthesis, run an ultracentrifugation, work with proteins and DNA and RNA, run a high performance liquid chromatography, liposome creation. I know this list is very long, but basically a lot of things. And so I was eternally grateful for this rotation because I got to learn an entirely new set of skills that I don't get to use in geology. And then this is also suggested was to talk about how this fits in with my thesis. In case you haven't seen me give a talk before, I'm usually thinking about early Earth and early Mars, specifically surface lakes on early Earth and early Mars, and how we could get the components for life geochemically. So thinking about the erosion of rocks on Mars, getting that into an evaporative lake setting and whether or not you can actually get prebiotic synthesis in those types of lakes. But it kind of stops at pure chemistry, and I've never gotten to take it past that and think more about biochemistry and prebiotic synthesis. So I think this project was really cool to take that next step and thinking about farther down the line, as well as not thinking about wet-dry cycling, but in the context of freeze-thaw cycling. And then this was sort of just fun pictures from Elsie. One thing that I was like really grateful to see was the Oshiki Festival, which happened in like my second week I showed up, and everyone's like, you guys, you have to go to the Oshiki Festival, it's this big deal. And I was like, why? And then I was sent this woodblock print from 1864 of the Oshiki Festival, and they were like, this is why we have to go. And I was like, okay, I'll go. And you go, and there's like thousands, hundreds of thousands of people who go to this festival. And my fun fact was that I took this train line, the Ikigami line, every day to work because I lived a few train stops away from campus. And that Ikigami line was literally built to take people to this festival in like after the Great Kanto earthquake destroyed any streets that were able to take you there. And so I got to ride the Ikigami line to the festival to attend Oshiki, and it was very cool. And it was very fun to be there with Elsie students. And then the last thing was like, I also got to celebrate Christmas in Japan. It turns out there's a whole set of traditions around Christmas in Japan, including lots of fried chicken. So I spent 6 hours cooking fried chicken for students during the Christmas party. And yeah, it was a good time. So I'm very happy for the rotation. And any questions?
Thanks so much, Kim. All right, got questions?
I was talking about like, what is it like to do science in Japan? Because like culturally different, like does doing science there feel different?
Yeah.
How do you approach it differently?
Obviously I can like only speak to like the one institution I was at and all that, but I think that at least in this group I worked with, like Tomoaki Matsuda's group, there was a lot of structure. And that's also partly because there's so many things at play here. It's also because I was in like biochemistry, and biochemistry runs so differently than geology. But it felt like there was a lot of structure around like how research is to proceed, and sort of like not taking the next step until there were reasonable results. Like something my advisor kept saying to me was like, don't do the next experiment until you have a publishable figure on the first experiment. And like kind of just a lot of structure on that. But I don't know, big takeaways about research in Japan, I'm not sure. That's hard to say. Any other questions? Yeah, so,
You said that it's the fast freezing, slow cooling, the slow thawing that had like the greatest.
yeah, the greatest encapsulation, yeah.
Are there particular environments that would be like conducive to this?
Yes, so this is something we were thinking about a lot because one of the other, she's actually a postdoc, but Natsumi Noda was trying to replicate actual conditions you'd see on like Europa or Enceladus in terms of very slow freezing and thawing rates. And those are kind of hard to replicate in a lab because they are so slow. But this is interesting that we saw the best encapsulation with the slow thaw, because this was actually the environmental condition, was 1 Kelvin per minute, that it's very hard to exceed that naturally. The problem is the fast freezing, right? Because we're seeing the best encapsulation with fast freezing. And so you'd have to think of an environmental condition where you're going to get a sudden freeze, but then a very slow thaw. And I think that's potentially like possible in certain environments, but it's maybe not most realistic. Yeah.
Yeah, can you help me to understand what is controlling the size of the particles that we are making in the lab, like what kind of physics or chemistry? Because I think I remember you showed some slide about like this droplet size, and I saw some Gaussians.
Yes,
So I'm
yeah, all the
curious,
like
like,
DLS measurements.
Yeah, I'm curious what is selecting the size.
Yeah, so basically you can make many different size LUVs. It's just dependent on your experimental protocol. And so the way we did it is if you create a lipid film and you concentrate it onto the surface of a glass vial, and then you redissolve it into solution, you're going to get, it's something like 1,000 to 10,000 nanometer vesicles automatically. And then you have to go through a process of actually extruding it through a filter to get it down to that 100 nanometer size you want. And so you can use different filter sizes to get them, like that's how we also got 800, is you just swap out to a different filter size. But that's just for LUVs. If you want to make the giant ones, it's actually an entirely different like experimental protocol.
So naturally, it's more like a uniform distributions, but you pass it through a filter to get what the size you want.
Yeah, because we're trying to just see one specific size.
Okay.
Yeah.
It strikes me that the fast freeze, slow thaw is the kind of thing we typically do in biology just to preserve activity of enzymes or structure of enzymes or that kind of thing. Do you think it's possible that what you're seeing is less the actual encapsulation and more the successful maintenance of an active enzyme or something like that in the middle?
Yeah, that's definitely part of it. Like I think I said on there a few slides back, I tried a fast thaw at 15C and everything died, right? Like as you would expect. And so that was like useful to do because like, yeah, you don't actually expect enzymes to survive that. But so yeah, you're right that there's some aspect of like with the T7 polymerase, it probably wouldn't function at high temperatures, at those like fast thawing temperatures. But I guess the other part of it is that because you're seeing that, that's probably something that was selected for over time, right? Like that we've ended up with these enzymes that have a preferential range. And so maybe it is because of how they formed. Now I'm just like spitballing, but.
So like, how important is it that, like, because you found that under several freeze-thaw iterations that there was still encapsulation happening, it just wasn't like the top result. So is that still an interesting result in itself? Like, did you expect them all to work under different freeze-thaw conditions? Or like, why were you looking for the best one instead of just the presence of that happening?
Sorry, do you mean with the type of condition or the
Yeah.
number of cycles? The
Like the type of condition.
Yeah, so, well, especially with IVT, it's just because we didn't see a lot of encapsulation, so we were trying, because it's like not quite substantial enough to think we're truly encapsulating something, which is
Okay.
why we tried other conditions. With calcine, we saw distinct encapsulation, but we also were just curious, like, how much can you encapsulate? Because naturally, in natural conditions, you're not gonna get as high lipid concentrations. You're not gonna have these perfect lab conditions to have this happen. So we want to make sure that it's viable and that you're seeing ridiculous encapsulation in the lab so that it might possibly happen in nature, right? Yeah.
Yeah, I guess like another question I have is when you're doing synthetic biology experiment, do you need to do anything to prevent the contaminations of like just earth biology? Like how do you kill the microbes around? Like,
Yes, I luckily didn't have to do anything with bacteria because those guys had to be like much cleaner than me. But there was just general protocol around like you have to always wipe down your bench, you need to be having your proper PPE, you need to have gloves on. And then when you're storing your samples, you have to make sure that, you know, there's not any contamination when you're putting it away in the freezer, when you're taking it out, like kind of just general things, but not like crazy. Like it was a lot more intense in terms of contamination if you're working with microbes, but these were sort of simplistic systems that weren't as sensitive.
I guess I'm asking like, could others just airborne microbes just land on your setup and then do their things and mess up your result? Is it a problem?
I guess, yes, but like at least with like calcium encapsulation, we're measuring something so simple that like if a bacteria ends up in our Eppendorf, it's not gonna increase the, well, I guess I shouldn't say anything like that. Maybe it's some random bacteria will artificially increase the fluorescence of calcium, but like other than that, I don't think that's supposed to happen, yeah.
Does Oshiki celebrate?
Yeah, so I have my like blurb on it, but it's in my notes, so I can't actually see it. But basically, it's a Buddhist procession. A famous Buddhist monk died on a big like what do you call it? Procession from Kamakura to Edo hundreds of years ago, and he died in Ikegami, and so they celebrate it every year. Yeah.
How do you say astrobiology in Japanese?
Do you know? I don't know. But yeah, that's a good question. Or like planetary science?
Yeah.
Okay, sorry, I'm putting you on the spot. I don't know, sorry. Well, thanks,
All right,
guys.
well, yeah, well, we go, thank Kim again for a great talk.